So friends, first we want to know what is DNA replication. So DNA replication is a process by which a new DNA is synthesized from another DNA. Now I am going to tell you the different type of enzymes involved in replication. A number of enzymes and protein takes part in autocatalysis reaction of DNA synthesis. These are together called replisomes. So the first enzymes is DNA polymerase. Three types of DNA polymerase are important. In short, DNA polymerase is called DNA pol. The first type of DNA polymerase is DNA pol 1 or DNA polymerase 1. This is synthesized from pol A gene and is monomeric type. The enzyme was identified by Arthur Kornberg in 1955 and is formed large in number. Now, the function of DNA polymerase 1 or DNA pol 1. It serves the following function. First function is polymerization activity in 5' to 3' direction but in slow rate. The second function is it has 3' to 5' exonuclease activity that helps in proofreading, also called DNA editing. The third function is it has 5' to 3' exonuclease activity also that removes Okazaki fragments from lagging strand. Now the second DNA pol is DNA pol 2 or DNA polymerase 2. This enzyme is synthesized by pol B gene. This was identified by Gefter, Niffer and Richardson in 1970. The function of this enzyme is less significant that takes part in DNA replication in necessary. Now the third type of DNA polymerase or DNA pol 3. This is the principal polymerase enzyme identified by Gefter and Tom Kornberg in 1971. Now function of this DNA pol 3 or DNA polymerase 3. The first function is it has polymerization activity in 5' to 3' direction. The second function of DNA pol 3 is it has exonuclease activities in 3' to 5' direction that helps in proofreading. It reduces chances of error, giving high fidelity. Next enzyme is DNA primase. The DNA primase enzyme is a specific RNA pol encoded by specific gene DNA G in E. coli. Now the function of DNA primase is this enzyme involves in synthesis of RNA primer. What is RNA primer? RNA primer initiates DNA replication. So the DNA primase is very important enzyme. Next enzyme is helicase. The helicase is very much important synthesized by DNA B gene in E. coli. This enzyme removes helix by unwelding it using ATP. Second function is it forms replication fork by breaking hydrogen bonds. The next enzyme is Gyrase. This enzyme is tetramer formed 2 identical alpha chains and 2 identical beta chains. Alpha subunits are encoded by Gyrase gene and the beta subunits by Gyrase gene. Now the function of Gyrase enzyme is this enzyme removes positive supercoiling occurs during unwinding of double helix. This enzyme has a nicking closing mechanism. The next enzyme is Topo Isomerase. In prokaryote, a circular DNA is present. Initiation of replication of this DNA requires a special enzyme that removes its supercoil found in relaxed condition. Two types of such enzyme has been identified that alter the topological version. These are Topoisomerase 1 and Topoisomerase 2. Topoisomerase 1 produce single strand break and remove single supercoil at a time, whereas Topoisomerase 2 produce two breaks and removes two supercoil at a time. Next enzyme is telomerase. This enzyme is formed in eukaryotic cell but essentially not in all eukaryotic cells. This enzyme contains a self-within RNA primer template and a randomly repeated sequence, that is kata TTAGGG in human. Function of this enzyme is it involves in replication of telomeric DNA and prevents shortening of DNA length in each replication step. Inactivity or absence of this enzyme in a cell tends its death. This enzyme is now considered as reason of aging. It is named as molecular clock. The seventh enzyme is ligase. This enzyme involves in joining ligation of two DNA segments. It uses energy from ATP and NAD as cofactor. So the function of this ligase enzyme is the Okazaki fragments after removals a new strand of DNA synthesized by DNA poly1 and joined by DNA ligase. So these are the enzymes which helps in process of DNA replication. That's for today. Thank you. Teaching is really fun.